Thank you, ma'am. Good afternoon, everyone. I'm Rajesh. I'm Rajeshwari, Assistant Professor, Department of Chemistry, Annai Violet Arts and Science College. It is my great honor and pleasure to welcome Dr. N. Rajendran and introduce him to you this afternoon. We appreciate his willingness to share his expertise and personal experience here. Let us share with you a little introduction about our guest, Dr. N. Rajendran. He is working as an Associate Professor, Department of Polymer Science, University of Madras, Chennai. His area of specialization is polymer nanocomposites, nanomaterials, synthesis of pH and photothermosensitive polymer and hydrogels. He is a life member of Chemical Research Society of India and the Society for Polymer Science. He has successfully completed 3 projects. He has published many papers in both national and international level. He guided 7 PhD and 6 MPhil candidates. Furthermore, he served as a deputy coordinator in GNR Instrumentation Center since 2022 till date. And he served as a doctoral committee member, PhD Viva was examiner, question paper setter, subject expert, selection committee member for the various colleges and universities. We once again welcome you, sir. We would like to take over the session further.
Okay, ma'am. Thank you, ma'am. Hello, sir.
Sir, screen share ஆயிடுச்சு சார். நீங்க ஸ்லைட் ஷோ ஸ்டார்ட் பண்ணிட்டு நீங்க பிரசன்ட் பண்ணிடலாம் சார். ஸ்லைட் ஷோ ஸ்டார்ட் பண்ணிடுங்க சார். ஓகே. கீழ பாட்டம் எஸ். கீழ பாட்டம்.
Okay, sir.
Yes, sir. Thank you.
Very good afternoon to one and all. First of all, I should thank Dr. L. Premila and Annai Violet College administrative staff for giving this opportunity. And I am going to present my research field, recent development on the synthesis of macromolecules stabilized noble metal nanostructures and their chemical and biological applications. Okay, introduction. So nanoscience. What is nanoscience? Nanoscience is the study of phenomena and manipulation of materials at atomic, molecular and macromolecular scales, in which at least one dimension should be less than 100 nanometer. That is, any particles having size less than 100 nanometer, that is we call the nanoparticles. And the nanotechnology is the creation of functional materials and devices and system through controlled manner on the nanometer length scales, and the exploitation of novel properties such as physical, chemical and biological at this length scale. This slide shows the materials that is exist in the natural as well as naturals in their different sizes, where the polymeric micelles and magnetic nanoparticles, polymeric micelles, polyelectrolytes, liposomes and magnetic nanoparticles are lying in the range of 10 to 100 nanometers. So nanoscience, even though nanoscience is the it is latest development, it is not a latest developed field, and we have some evidence for existence of these nanomaterials in the ancient stage also, ancient age. That is, for example, the liquor gas cup. This cup is coated on the silver and gold nanoparticle over the surface. Then upon illumination of light, it exhibits as a green and red colors. Still now it exists in the British Museum. The another example is the North Rose windows in Notre Dame Cathedral, where the design is made upon nano-coated materials. And the other one is Damascus quad, where when you magnifying this quad, it will exhibit the springes, showing that this the material is made upon nanoparticles. And also one of the authentic evidence the existence of nanoparticles in a chemical aspect, that is the gold nanoparticles. It was prepared by Faraday in the year 1857 by using the aurichloride with the phosphate salts, and the particles are reduced at the nanometer. But in those days, he don't have any sophisticated instrument, but he called as a nanocolloids. Still now, the solution is adjust the same colors without any pad. So this slide shows why the nanoparticles are so different colors and so important. First of all, the one of the major properties is the surface to volume ratio. That is, if the particle is reduced to smaller size, the physical and chemical properties are entirely different. Okay, so here you see that in this slide, here you say that the gold in the bulk form is the yellow color. We know that, and similarly, silver is the colorless. Once if you reduce the particles in their different range, different size, that is in the range of 100 to 1 to 100 nanometers, that depends on the size of the particles as well as the shape. The solutions exhibit different colors. Why it is so? For example, in inorganic complex, the complex exhibit different colors. That is due to the either charge transfer or metal to ligand or DD transition like that. Whereas in this case, we don't have any ligands. Just we are reducing the metal particles from the higher oxidation state to lower oxidation state. After the reduction, all the metal nanoparticles are in the zero oxidation state. And also we don't have any ligands around the metals. But still it exists as a color. Why it is? That is we call the surface plasmon resonance. Because when you reduce the particles in a smaller size, what will happen at this length scale? It becomes a very small. So when it passes the light on the particles, the outermost conductance electrons will oscillation. Okay, so because of these oscillations, it needs some energy that will take from the either in the UV or laser light, so that accordingly the color is reflecting. That is why the metal nanoparticles, if you reduce it in a different range, that will exhibit different colors. That is the one of the peculiar properties for the metal nanoparticles. So from the UV visible spectra, that is the very basic technique we can able to visualize under naked eye, as well as in the UV visible spectra we can find out the surface plasmon resonance. So based on this, we can come on conclusions whether the prepared particles are in a spherical shape or anisotropic nature or some other size. The next one is the metal nanoclusters. That is, surface plasmon resonance is the coherent oscillation of conductance electrons induced by interactions with the electromagnetic field. This is called the surface plasmon resonance. The next category of the metal is the noble metal nanoclusters. That is the recent findings within the 10 years. So why we are called as a clusters? That is, any particles having size less than 2 nanometer. Less than 2 nanometer or maximum 5 nanometer. If the less than 5 nanometer, within this range, the properties of the metal nanocluster is entirely different from the nanoparticles. In what way it is different means, in the metal nanoparticles, it will exhibit a surface plasmon resonance. Whereas in the nanoclusters, the same metals, because of the existence of size in the range between below 5 nanometers, the physical and chemical properties are entirely different. In what way means, metal nanoparticles are exhibit surface plasmon resonance, whereas the nanoclusters were exhibiting a fluorescent properties. Why it is a fluorescent properties? Means in the below 5 nanometer size, below less than 5 nanometers, these metal nanoclusters are behave as a molecular-like properties. That is only a few atoms are joined together. That is we call the cluster. If you measure the size, that will exhibit below 5 nanometer. Because of the less number of atoms are attached with each other, it behaves as a molecular-like behavior, and the energy level also in a discrete manner, like molecules. Whereas in the nanoparticles, it is being a bigger size, so that the energy levels are merged with each other. Because of this energy level difference, the nanoparticle is entirely different from the nanoclusters. Okay. Normally, nanoparticles we can synthesize top down as well as a bottom up approach. That is, in the top-down approach is the physical methods. That is mechanical subdivision of metallic aggregates. Whereas in the bottom-up approach, chemical method, that is the nucleations or growth of the metallic atoms. Whereas in the top-down method, this approach involves starting of black bulk materials and designing or milling it desired shape. This technique is similar to approach used in the semiconductor industry for forming devices. However, the method used for in this approach is not practiced because it is very hard and it needs a sophisticated instrument. The another drawback is getting a uniform sized particle is very difficult in these physical methods. So that the bottom-up method is having some disadvantages. Whereas, the top-down approach. Whereas, the bottom-up approach is the chemical methods, that is self-assembling, doing the growth stage, or when you reacting the metal nanoparticle, when you prepare the metal nanoparticles, or these metal nanoparticles are joined together and it for growth process. So we can control the size and the shape of the particles at any point. So as a result, the prepared particles will be in the uniform size because of the atom by atom or molecular or cluster by cluster association. So as a end of the reactions, you will get the uniform size nanoparticles. Whereas the atomic metal elements are made up of nanoparticle by induced aggregations, yielding uniform particles with controlled definite size, and protective coats is needed for the stabilization of metal nanoparticles. However, it's a need protecting coating as well as the reducing reagent. The advantage of the chemical reduction method is it is used to control the size and shape of the transition metal nanoparticles, and it is easy to implement under simple and mild conditions. Whereas in the physical methods, it is very difficult to prepare a metal nanoparticle because we need a sophisticated instrument. Whereas in the chemical methods, even in the lab conditions, simple way, we need not require sophisticated things. Simply we have some glasswares and metal salts and other reducing and stabilizing agent. Those things are enough to carry out the reactions. And also prepare in a large scale a wide range of metal nanoparticles like silver, gold, platinum, ruthenium, copper can be prepared by this approach. And there are, in the chemical methods, there are three constants mainly required. That is the metal salts, reducing reagent, and stabilizing reagent. Normally, metal salt, if you want to prepare a silver nanoparticles, you have to take silver nitrate or silver chloride like that. And the metal gold nanoparticles, auric chloride, platinum, ruthenium, that respective metal salts we need to take. And we have to use the reducing reagent. Why it is reducing reagent means after the reductions in this metal, here the oxidation state has to be reduced to zero oxidation state. For this purpose, we need to add reducing reagent. That is the wide range of sodium borohydride, hydrogen, hydroxylamine, ethanol, ethylene glycol, and polyaniline, and a number of there are number of reducing reagents available. So with those things we can use for reduction of metal salts. And the other one is the stabilizing reagent, where the role of stabilizing reagent is to control the metal nanoparticles in the desired shape. Because once if you reduce the metal salt in the zero oxidation state, what will happen? The particles are continuously growing. So that we need to arrest the particles in a smaller size, for which we need to add some stabilizing agent like ligands, surfactant, polymers, phase transfer catalyst, and dendrimers. So this is one of the examples for the preparation of metal nanoparticles using the surfactant, where here they used for CTAB. That is, CTAB is the cationic surfactant, cetyl trimethyl ammonium bromide, that is the stabilizing reagent, and they add sodium borohydride in silver nitrate. So after the reductions, what will happen? So the CTABs were covered over the surface of the particles, so that in that manner the particles are stabilized or control the growth of the size. So after the preparations, we need to confirm the nanoparticle synthesis by using UV full spectroscopy, where this one is the surface plasmon resonance, that is surface plasmon resonance, where at the different time intervals, as the reaction is processed, the SPR band around 420 nanometer is increasing. That is the one of the characteristics peak for the silver nanoparticles. And also during the preparation itself, we can able to confirm this slide. You see that the solutions initially has the colorless, whereas after the reductions, the solutions exhibit intense red color solutions. So from this we can conclude that the prepared solution, conclude that the existence of silver nanoclusters. And further we can confirm by term transmission electron microscope and DLS for the size and the shape. And this is the example for the another approach, different stabilizing agent, that is the dendrimer, polyamide amide dendrimer and glycodendrimers, where the dendrimer is it's macromolecules, it's entirely different from the polymers. In the polymers, we have a range of molecular weight in a solution, whereas in the dendrimers, all the molecules are in the uniform size. So that there are three regions, core and core, void, and branching unit. Because of this template, it acts as a very good template. Because of this scaffold, it acts as a very good template for the stabilization of metal nanoparticles. So when you add the metal salts initially, because the attractions between the functional groups with the metal salts it is embedded over on the topologies, and once you add the sodium borohydrate, these metal nanoparticles are reduced in the metal salts are reduced into metal nanoparticles. This is another example for the polyethylene imine. Yeah, this is the one of the hyperbranched polymers we can use as a stabilizing agent for the stabilization of metal nanoparticles. Here they used for the encapsulation of silver nanoparticle using polyethylene imine, and after the reductions they confirmed by transmission electron microscope as well as DLS. And after the formation of this, they used as bacterial growth, antibacterial activity. Here you see that the nanoparticle coated areas are control the growth of the bacteria over the uncontrolled, uncoated surface. So even though there are many protocols available for the there are a number of protocols so far available for the synthesis of metal nanoparticles, metal nanoparticle using chemical method. The resulting for the control preparation of uniform size particle is very difficult. Even though if you optimize the either the concentration of stabilizing agent, reducing agent, and other reaction conditions, getting uniform size is very difficult. So that in the year 2006, C.J. Murphy, he is one of the famous scientists. What he did, he used the seed-mediated approach for the synthesis of metal nanoparticles. The other thing is, in the just ordinary chemical methods, just by adding up metal salt, surfactant, and reducing agent, mostly these particles are in the spherical in nature, spherical in shape, where it showed the SPR band, if it is a silver 420 and gold nanoparticles 520 like this. But in the preparation of anisotropic particle is very difficult. So that in the year 2006, C.J. Murphy, what he did, he used them for a seed-mediated approach for the synthesis of anisotropic particles, for which he prepared two solutions. That is the seed solution. This one is the first step. He prepared a seed solution. And the second step is the growth solutions. Okay, in the seed solutions, he used respective salts. Here he used aurichloride, sodium citrate, that is a stabilizing agent, and added sodium borohydrate. That is a strong reducing reagent. And he prepared seed solutions. That is seed solution means the particle is very small size. And the second solutions, he prepared a growth solution. That is, he added aurichloride with acetyl trimethyl ammonium bromide surfactant, and where he added ascorbic acid for the preparation of growth solutions. So he add a mild reducing reagent. So by mixing the seed solutions and as well as the growth solutions, by mixing the seed as well as the growth solutions, you will get the long nano rods. So that is, the formed particles are in anisotropic nature. How it forms means the seed tab, we know that it's at the CMC it exhibits in spherical in nature. Okay. So upon increasing the above the CMC, what will happen? So this spherical in shape of the surfactants are joined together in the form of cylinder, where in the interior size of the interior size, it provides a site for the growth of the nanoparticle, so that when you add the seed solutions and the growth solutions, these metal ions are reduced inside the template and it forms a rod shaped particles. So further we can confirm by transmission electron microscope, where it shows the synthesized particles are all are uniformized in the rod shape. So in the ordinary chemicals, it is methods, it is very difficult. Whereas in this approach, we can prepare most of the materials or in the metal nanoparticles or in the rod shape. Similarly, we can by optimizing the conditions and all those things, we can able to prepare the rod shaped, diamond shaped, and the anisotropic shaped metal nanoparticles. We can able to prepare. Actually, this report was made by Wang in the year 2006 itself. And in the chemical method, normally they used for reagents or even though it's toxic, definitely it's less toxic, and using the reducing reagents also toxic. So after the reductions, it is very difficult to prepare or separate the reducing reagent. That will be exist in the solution only, because it is very difficult if it is water and other forms. So that when you go for biological or other applications, so it really it is so it will give some complications. So so because of these issues later the scientists they also tried to another approach for the synthesis of metal nanoparticle using the plant extract and fungus and plant extract, bacteria and fungus and other biological origin source as a reducing and stabilizing agent. And in the year 2005 and 2003, Murali Shastri is one of the famous scientists in India. He reported number of synthesized number of nanoparticles using different plant extract for the purpose of biological applications. So this report shows the synthesis of metal nanoparticles using tamarind and neem leaf extract. So just boiling the leaf extract, and he used for synthesis of metal nanoparticles. You see that here upon changing the amount of plant extract, we can able to prepare anisotropic metal nanoparticles. Here you see that these nanoparticles are either in the hexagonal or triangular shape or pentagonal bipyramidal shapes. So in the UV, in the usual absorption spectrum, in addition to the formation of SPR band around 500 to 600, another intense band appeared in the NIR region. That is the characteristics of the existence of anisotropic nanoparticles. Similarly, silver nanoparticles also prepared using this plant extract. The other reports used olive oil extract. Olive oil extract, we know that mostly they used for medical purpose, and he prepared silver and gold nanoparticles. Here, this is the silver nanoparticle. This one is the gold nanoparticles, having different anisotropic nature with different sizes. So we can you see that depends on the amount of solutions, that is the extract, we can able to different sized metal nanoparticles. Accordingly, the SPR band is shifted from the around 500 to 1000 to 250. From that we can conclude, and further 10 measurements it shows a different size. And based on this, we also synthesized in the year 2009 reported silver and gold nanoparticles using pilantine as a reducing reagent. And we got, interestingly, we got some silver gold nanoparticles with a different anisotropic in nature, hexagonal and triangular shape. That is depends on the plant extract, amount of plant extract added into the solutions. And also in the silver nanoparticles are nearly spherical in shape particles. Be confirmed by FDA. This is the proposed things, the pilantine, because in the pilantine and plant extract, pilantine major constituent is the pilantines. That may be the responsible for the reduction of metal ions and stabilization of it. And also we have reported silver and gold nanoparticles using apine as a reducing reagent, and confirmed by the existence of anisotropic gold nanoparticles and the silver nanoparticles were confirmed by the transmission electron microscope. And solution also exhibited different colors, which showing that upon changing the plant extract, that is the amount of extract, we can able to prepare different sized metal nanoparticles. Even though plant extract is the that is low toxic or biocompatible nature for the preparation of metal nanoparticles, the drawback is once if you prepare the metal nanoparticles using the plant extract, the stability is question mark. That is mostly these metal nanoparticles are stable up to 1 week or 2 weeks only. Okay, if you go for applications or long-term other purpose, it should be stable, but the stability is the problems. And the other thing is um, there are in the plant extract there are a lot of constitute. Okay, so that we cannot exactly say which constitute is responsible for the reduction of metal ions. That is also the problems. The third thing is reproducibility, because when you go for a different time interval, if you want to prepare the nanoparticle using plant extract, you cannot get the exact concentration. That is also the problem. So reproducibility also a problem in the plant extract. So because of these issues, researchers and scientists, they are moved to prepare a biocompatible biocompatible chemicals. In that way, I have used bile acid as the biosurfactant. Biosurfactant. This report shows the preparation of silver and metal nanoparticles using sodium cholate and sodium taurocolate as the reagent. This is the first report. First time we are reporting this result, synthesized this compounds. So after this, upon changing the concentration of the bile acid, so conjugated bile acid conjugate amino acid conjugated bile salts, it shows a different colors. So that we confirmed by transmission electron microscope and transmission electron by XRD and DLS UV visible spectroscopy. Why the bile acid is so important means bile acid is because in traditional surfactant there are many reports available using sodium silver for the synthesis of silver and gold nanoparticles, but bile acids even bile acids no report no one has reported. The first time we reported in the year 2009, and subsequently we made lot of report on this using this. So why it is so important means bile salts are natural. The another thing is in the ordinary surfactant, either CTAB or SDS or Triton X100, we need to add some external reducing reagent. Whereas in the bile acids, we need not add any external reducing agent because of the availability of some reducing functionality group that will responsible for the reduction of metal salts. Why it is so important means bile salts are naturally occurring steroid detergent members, which aids the digestion of fats and lipids. They are amphiphilic in nature. The interior of bile salt micelles are more rigid and complex than micelles formed by the conventional surfactant. The interaction of bile salt with nanoparticle is different from the traditional surfactant. Furthermore, the chemically different functional group, unique ample release stream, easily available and low cost, make them ideal building block for the stabilization of metal nanoparticles. Because of this reason, we use the bile salts as a stabilizing as well as the reducing the agent and reducing the agent. In the bile acids, actually the structure is shown here. Here you see that there are depends on the OH. It can be classified cholic acid, lithocholic, and deoxycholic acids. And here there are two domains: the hydrophobic as well as the hydrophilic region. So this forms two different types of the micelles. That is the primary micelles and the secondary micelles, where the primary micelle is driven by the hydrophobic interactions, whereas in the secondary micelle is formed by the hydrogen bonding. So from the after the formation of micelles, we can use this material as a template for the preparation of metal nanoparticles. So this slide shows the synthesis of sodium cholate. Synthesis of gold nanoparticles in a different size and shape using sodium cholate as a reducing and stabilizing agent. And here you see that from the UV visible spectra, upon increasing the concentrations at low concentrations, it forms anisotropic nanoparticles, whereas at higher concentrations, it forms a spherical nature. Why it is so means, since it behave as a strong reducing as well as a stabilizing agent, what will happen at low concentration, the reducing functionality is very low, because it reduces the auric chloride or gold chloride in a slow manner. Because of this slow reductions, what will happen, the reduced particles are having enough time to arrange in a particular shape. So that at low concentrations, it favors for the formation of anisotropic nanoparticles, whereas at higher concentrations, the reducing functionality is very high, the reduction process is very high, so that the particles are mostly in the spherical nature. So these things are confirmed by FTIR and DLS measurements. And after the reductions, this solution was after the optimization we used for the detection of geminiviral DNA using colorimetric technique. That is because this nanoparticles are very much sensitive to the analytes. That is, analyte means if you add any interference of ions and other things, that will the color will changes. So because by using these advantages, we use it as a colorimetric probe for the detection of geminiviral DNA. So normally instead of going the PCR and other costly equipments, just colorimetrically we can able to identify how much the probe or single stranded DNA getting degeneracy probe are getting sequenced. So from that we can from the UV spectra, here you see that initially the prepared solution is the red color. So after the additions, it becomes increasing the complementary. What will happen? So that is the blue color. Why it is so means the single stranded DNA. What will happen? So it cover over the nanoparticles, so that the particles are getting stabilized. Here you see that the picture is schematically represented at after the addition of the single stranded DNA. What will happen? So this single stranded DNA, it covers over the surface of the nanoparticles. It won't allow for the aggregations. However, after the addition of double stranded DNA after the sequence, what will happen? So the single stranded DNA removed from the surface, as the solution is getting aggregations. So from this color changes from the red color to blue color, we can able to identify colorimetrically for hybridizations. And also further we confirmed by the transmission electron microscope. Actually, this paper was published in the Journal of Physical Chemistry in the year 2012. And and also we have a synthesized silver nanoparticle using sodium cholate, where depends on the concentration of the sodium cholate, we can able to prepare different sized nanoparticles. Accordingly, the color of the solution changed from the light yellow to purple, light yellow to brown colors, and then it's padded. So that from that we can able to optimize, and further we can confirmed by the transmission electron microscope, where prepared nanoparticles are showing less than 10 nearly 10 nanometer size. This is the schematic representation how this is the mechanism of how the sodium cholate reduce the silver salt in below and above the CMC. In below the CMC, you see that mostly the formed particles are in anisotropic in nature, whereas above the CMC, the particles are in spherical in shape. So after the synthesis, the silver nanoparticle be used for the galvanometric detection of metal ions. That is metal ions. If you add toxic metal or heavy metal ions, what will happen? Here we have only two different ions interacting with the metal nanoparticles compared to other metal ions, so that we can use as a selective for the detection of colorimetric probe. For the detection of colorimetrically, we can detect the metal ions in the environmental samples at the nanomolar level. And also these metal nanoparticles are very good catalysts. So in order to check the catalytic efficiency of the nanoparticles, we demonstrated both spherical and anisotropic nanoparticles, where it shows the anisotropic nanoparticles are greater the catalytic activity over the spherical inside. So after the synthesis, what we did, we kept the solutions in the room temperature, and after 6 weeks, we kept the after the 6 weeks, we monitor the solution in the UV light. So surprisingly, what we got some emission properties. So emission properties, really it's a very good interesting results. And what happens once the formed metal nanoparticles again it converted into a smaller sizing through etching process, so that the bigger size metal nanoparticles are again converted to a smaller size, that is the clusters. These clusters where it was confirmed by the emission properties. Here you see that this is the greenish bluish green emission properties. This is the UV light, in presence of UV light and daylight, whereas in the daylight it shows a red color. The same solutions when it is a UV light, it shows a bluish green emission properties. So it's showing that the existence of or formation of clusters. And further, we separate the nanoparticles, existence of both nanoparticle as well as the cluster. We separate the nanoparticles from the cluster by adding the sodium chloride. So what will happen is the bigger size nanoparticles are getting settled in the bottom of the vessels due to the salt effect, and the resulting top solutions are emission properties. Only it shows here. You see that there is no, after the addition of sodium chloride, there is no any color on the solutions. Solution, that is all the metal nanoparticles are deposited on the bottom. So the resulting solutions, in that way we can able to prepare metal nanoparticles from the metal nanoclusters. And after that we characterized using UV spectrum. As I mentioned earlier, the metal nanoparticles was the SPR band. Here you see that there is a SPR band around the 520 nanometers, and could be due to the existence of red colors. After the addition of sodium chloride, there is no peak in this region, showing that the metal nanoparticles are completely separated from these solutions. So that the nanoclusters, we don't have any SPR band, nanoclusters. So no peak, up to 300 we don't have a peak, and also below which we have some step-like features, that is the characteristics of the nanoclusters. And further we confirmed by transmission electron microscope, and the particles here you DLS, you see that this particle is nearly 1.3 nanometers. So such a smaller sized nanoparticles is it's entirely different from the nanoparticles, and the physical and chemical properties also different. And from the emission spectra, upon excitation at 365 nanometer, it emit at 478 with mono exponential, and further confirmed by time correlation single photon countings, where it shows only one emitting species. And being in fluorescent emission properties, this nanoparticle was used as a probe for the bioimaging. We can use for biological applications, because in bioimaging and other applications, mostly they will do used for organic fluorophores, where the organic fluorophore and semiconductor quantum dots and semiconductor quantum dots and organic fluorophores. Those fluorophores are really, it's a very toxic and highly potent bleaching. Okay, so that we studied first time, we have used them, whether it is whether we can use as a bioimaging studies, used as a probe for bioimaging studies. We checked, so that we studied for before the studies for other biological applications, we need to study for some toxicity studies, for which we used for zebrafish as a toxicity studies. Why we choose means, if you go for rat and other animals, we have ethical issues. So because to avoid those complications, we did simply by using the zebrafish as a in vivo model system. Why we choose as a zebrafish as a model system means it close homology with human genome, and embryonic development is completed within 120 hours, and with well-defined developmental stages, transparent embryo and larval stage, inexpensive experimental setup, smaller size, and maintenance and other things is very easy. Because of these advantages, we studied, first time we studied zebrafish as an in vivo model system for using gold nanocluster as a fluorescent probe, where we used gold nanoclusters. Here you see that the blue in the embryos after the injection of nanoclusters, it completely absorbed on the embryos. At the different stages, we monitored. At the end of the growth, we don't find any defects. So from this, we can conclude that the prepared nanoclusters, we don't have any defects. We conclude that the prepared nanoclusters is non-toxic. So from that, primarily we can whether it is. So this one shows the survey, that is the survival and post fertilization plot. Here you see that up to 150 microliter, up to 150 microliter is comparable to the control. So we can use such a probe up to 150 microliter for bioimaging studies. And here also the aging rates also, it's not that much of difference compared to one control up to 150. So we can use as a probe for the in place of semiconductor quantum dots and organic fluorescent probe. And similarly, we also synthesized silver nanoclusters using the sodium cholate template. Here you see that after 4 weeks, we got the same protocols, but we got blue emission properties, where the silver clusters source blue emission properties in the presence of UV light. And further we confirmed by fluorescence spectra, where it shows the excitation at 350 and the emission peak was observed at 460, around 460. And further we confirmed the existence of cluster compositions, and there are 2 types of compounds, that is Ag4 and Ag6. Further, from the multi-mass spectra, we confirmed that the existence of the cluster in the form of Ag4 and Ag6. Further, transmission electron microscope provides the size of the clusters, and the size is nearly less than 2 nanometer. And similarly, we also checked we checked the zebrafish for bioimaging studies. In this case, also we don't find any defects. We don't find any defects and up to 200 microliters. So that this paper was published in ACS Applied Materials. And next we have synthesized, and next we tried to synthesis for conjugation, some modifications of folic acids using some biocompatible compounds. So what we did, we used folic acids with we added different amino acids in order to increasing the physical and altering the physical and chemical properties. We introduced different biocompatible moieties around on the folic acids, because the micelles, formation of critical micellar concentrations, other properties are due to the hydrophobic as well as hydrophilic interactions. So that we introduce a different functionality on the folic acids through amidation chemistry, and we studied for nanoparticles and other biological applications. This scheme shows the synthesis of amino acids conjugated folic acids through acid amine coupling reactions. After the preparation of folic acids with this is the tryptophan, tyrosines, cysteine, and valine, and phenylalanines. These amino acids were used by the conjugation with the folic acids and optimized using the pH. Here you see that around 10 to 9 the silver nanoparticles are more stable. So this is the optimization as compared to highly basic and acidic medium. And in presence of sunlight, the tryptophan capped, this spectra shows the useful spectra of tryptophan capped silver nanoparticles under different conditions, where it shows in the sunlight irradiation shows high intensity SPR band as compared to other conditions. And after that, the size of the nanoparticles was studied both the transmission electron microscope as well as DLS, where the prepared nanoparticles are around, depends on the nature of the amino acids that will controls in the different scale. And further we confirmed by XPS measurements the existence of elements on the nanoclusters. And finally, we used for the antibacterial applications of applications for synthesized different amino acids. And next, and also we synthesized the N-choline amino acids for the synthesis of gold nanoparticles. Gold nanoparticle through the grain chemical approach, and after the synthesis we used for the colorimetric probe for the detection of metal ions. This is the protocol optimization protocol at different concentrations. The SPR band is shifted. So from this we optimize the exact concentrations, required concentration for the preparation of gold nanoparticles. So depends on the concentrations of the concentrations, it exhibits different colors. Similarly, the pH also we optimized at a different pH for these nanoparticles, where here it shows around 8 to 9. 9 is the optimum condition for all the cases for the stabilization of metal nanoparticles. Above and below it, the particles are unstable. Then after the synthesis, we characterize the transmission electron microscope and DLS array. So depends on the depends on the nature of the substrate. The existence of particles are from 13 to 80 nanometer, different sizes, range of sizes. And further we confirmed by XPS measurements, the XPS. And also colorimetric detection of colorimetrically we used, because of the after the optimizations we used as a colorimetric probe for the synthesis of gold nanoparticles. And after the addition of different metal ions, these gold nanoparticles are specifically detect the manganese, only manganese compared to other metal ions. And whereas in the case of tyrosine capped gold nanoparticles, it selectively quenches or detect the color changes occur only for the chromium ions as compared to other ions, and which was confirmed by both naked eyes as well as the UV spectroscopy, so that we can use as a colorimetric probe for the detection of ions. Whereas in the case of valene capped gold nanoparticles, it detect only detect cobalt ions, cobalt and nickel ions in the environmental samples at the nanomolar concentrations. And whereas in the case of phenylalanine, stabilized phenylencholine, it selectively detects the lead ions at the nanomolar. So from these color changes, we can confirm. This is the mechanism, proposed mechanism, how the metal ions selectively interact with the metal nanoparticles, how the color changes is occur. And also using this the encolile template, we prepared red emitting gold nanoclusters. So far in the previous case, unmodified one, we prepared only for blue emissions and bluish emission nanoclusters. Whereas in the amino acid conjugated folic acids, we prepared red emitting nanoclusters. This is the schematic representation. This is the protocol for the synthesis of red emissions. And the synthesis was optimized by using the fluorescence as well as the UV visible absorption spectra. Here the different ratios and different ratio of metal salt and encolile amino acids were used at a different ratio for the optimization of the optimization conditions. Here you see that the solution is both daylight as well as the UV light, where the UV light, the last solutions where it shows a red color. This is due to the formations of the that is the existence of the red emitting nanoclusters. And further we confirmed by the UV spectra, we don't have any surface plasma resonance in this range 500, and the peak where a small shoulder or weak shoulder step like features observed in the UV regions. The same solutions, when you measured in the emission spectra, it gives a intense emissions in the 650 nanometer. In 650 nanometer. So from that we can confirm the solution also it's a red emitting, so that this is the optimization so for the synthesis of and upon different excitations it shows a highest index peak at 400 nanometer. And from the lifetime measurements also the existence of one only one emitting species. The encolile system was further confirmed by transmission electron microscope, where you see that the particles are mostly with average of 2.3 nanometer in size. So after the synthesis, here the same things we also used for bioimaging applications in the zebrafish, and so showing that as compared to controls up to 25 micromole, we don't find any differences. So from that we can conclude that we can use as a probe for studies. And even this is also for encolysis for cell viability. Up to 15 micromole, there is no change, that much of change as compared to control. So we can use this. And in the in vivo, in the previous case is the in vitro, this one the in vivo. Here in the zebrafish, the growth of the zebrafish at the end of the growth, we don't find any defects. So in that, it's a biocompatible nature for up to 50 microliter for cysteine capped as well as encolysis cysteine capped gold nanoclusters. Then this is the encolysis cysteine capped gold nanoclusters. We don't have toxicities. Then further we functionalize the bile acids using thioline click chemistry approach, and further we modified by introduced different surface charges. We introduced the cationic surfactant, proposed cationic surfactant, because the cationic moieties are extensively used for antibacterial and other application cationic substance because of the interactions with the cell membrane. So that we prepared cationically bile acids, and this is the optimizations for detection of CMC using fluorescent probe, and the proposed how the interacting with in the CMC, how it assembled with each other. And from the some measurements, we confirmed the existence of assembly of fractal patterned cationic folic acids at different concentrations. And after this is the same microscope. Here you see that depends on the concentrations. The assemblies are different in a different mechanism. And also we studied for the antibacterial activity, biological studies. First we used different species and found out the antibacterial activity for different prepared compounds, and also the hemolytic activity also we studied, so that from this result it's showing that the compounds don't have any toxicities. So after the synthesis, we used for preparation of metal nanoparticles. So depends on the concentration of the metal salts, it showed different colors. And further we confirmed by SIM and transmission electron microscope and microscope. So and also hemolytic activities were studied by cationic gold nanoparticles in a different concentration, so that we can use as a it shows any toxicity, don't have any toxicity. And finally we used for A549 lung cancer cells for whether it is any anti-cancer activities. So we got some good results at the higher concentrations. These compounds were destroy the cancer cells. So from the fluorescent microscope, we confirmed this is the proposed mechanism how they interacting with the cancer cells. These are the publications we reported so far from these results. And finally, I thank once again thank the college authorities for giving this opportunity. If you have any questions, please raise.
Thank you very much, sir. You have filled our brain with your valuable content, sir. Let's start with the question and answer session.
Okay.
Participants can kindly post your questions in the chat box.
Sir, I have a question.
Yeah, tell me.
Sir, can we say that nanoparticles are always colloidal in nature?
Colloidal nature means we can say colloidal, but in those days they called colloidal. But nowadays It is not a colloidal. Colloidal means mostly it's a turbid solution. Okay, the particles are in the micrometer size, whereas in this case it's a transparent solution. The particles are nano size. So we nowadays they won't call any colloid. It's a nano solution or nanoparticle solution.
Okay, sir.
Yeah.
Sir, another question.
Yeah.
When we prepare nanoparticles of different sizes,
Yeah.
whether they have different properties,
Yeah,
depends
definitely.
on their size, sir.
Yeah,
Okay.
definitely. Because in, for example, gold nanoparticles, mostly in solutions, if you want to prepare at a normal procedure, it forms a spherical in shape. So if you want to adopt some special template and other conditions, we can prepare a triangular or hexagonal shape, as I told in the previous case. And so in the spherical shape, it observes SPR band at around 520 nanometer. Whereas in the triangular or hexagonal shape, the SPR band was observed at NIR region. So in the NIR, the materials, any materials having absorption in the NIR region, so that we can use as a as a material for the destroy of the cancer cells. Okay, so if it is NIR absorption means nowadays they are using for destruction of cancer cells. Okay, so for this purpose we can use the same things. If it is spherical in nature, we cannot use. So it depends on the shape and the size. The property is also different.
Sir, one more question, and it's final question.
Yeah.
If we are going to prepare nanoparticles using biomaterials from plant extracts,
Yeah.
do we need stabilizers to stabilize the nanoparticles, sir?
Not required.
Not required.
It act both reducing as well as stabilizing. The thing is, stability is very less. It will be stable only 10 days like that only. After that, it becomes getting deposited. And the other thing is reproducibility also problem, because in every time we cannot use same plant extract. We can use even plantain also in a different volume and amount we will take it. So it depends on the concentrations, reproducibility different, and also purifications and other problems we have. Yeah.
Yes, sir. Thank you, sir. Thank you.
Thank you, sir. Thank you so much, sir.
Thank you, ma'am.
On behalf of management of Annai Valley Arts and Science College, I extend a very hearty thanks to Dr. N. Rajendran for gracing this session and sharing with us your valuable content today. It is a very informative session. Thank you once again, sir.
Thank you, ma'am.